بسم الله الرحمن الرحيم. Morning, Walid.
السلام عليكم.
Salam. Good morning, Mu'taz.
Good morning يا حسن.
Okay, what are the others? Okay, good morning.
السلام عليكم.
Alaikum salam. Good morning, Hussein.
موني
Abdullah Al-Eid, good morning.
ترى العيد
Al-Eid.
انسر.
Okay, give him the chance to find himself.
طيب.
Good morning, Abdullah.
السلام عليكم.
Nasir.
هلا والله.
Nasir Sharif.
الو تسمعوني؟
صوتك بعيد.
Yeah, your voice is so far, but we at least you know that we are greeting you in the morning. Should I start the session now, dear students, or should I wait for like a moment, maybe others will come?
الو
Hi.
تسمعوني؟
Yeah. Okay. Your sound, there is something went wrong with your voice, but it's okay. It's okay. It's okay. It's okay.
We won't ask you.
What?
We won't ask any question for you.
سؤال لي
Rakan Al-Bays, Rakan Khalid.
Good morning Mr.
Morning, Rakun. Good morning, Khan.
عبد الله حاول انك ما تتكلم.
Khalid Al-Harbi.
هاي مسلم.
Khalid Ahmed. Good morning, Khalud.
خالد
Good morning مسلم.
Hello.
Al-Walid, yes, please. Yeah, that's great. Mute yourself. That's much better. I think Abdullah Al-Eid, mute or not mute, it's okay. For us, it's okay
Hello.
because your sound is so
Hello.
far, but it's okay.
You can hear me الحين؟
But your voice is so far, as if you are in the far distance. I don't know what happened.
عبدالله
ناو الحين
ما زال
الحين كويس؟
اسمعك اهم شيء.
The most important that you listen, you heard me. If you want something, just write it. Okay, write it in a message. I can manage it. No problem.
Okay.
Fahad Al-Awaji, welcome back. Fahad Awaji. Mr. Fir'aun. Fir'aun didn't use his mic till now. It's okay. I think I have now 8 participants. I may start my session now. What do you think, dear students?
Take your time.
Yeah. Okay, that's great. Okay, I think Haji is still sleeping till now. I don't know what happened to him. So while waiting for him to come, I'm just working. Then I make fast revision of what we studied the previous session. So if we want to, until maybe they are coming soon. Okay. Um, Actually, after Bismillah ar-Rahman ar-Rahim, I want to greet you all and say to you that I'm so proud of your attendance. Good morning, dear students. Then I want just to remind you about what we studied in the previous session about these three essential questions. What are the different tools and processes used in genetic engineering? How does genetic engineering manipulate recombinant DNA? What are the similarities between selective breeding and genetic engineering? How can genetic engineering and biotechnology be used in improve human life? And this is the main point. This is the most important point that I choose this lesson to teach you. So now the new words, what we had, and some of this we are going to continue with it. Genetic engineering, genome, restriction enzyme, gel electrophoresis, recombinant DNA, plasmid DNA ligase, transformation, cloning, polymerase chain reaction, and then transgenic organism. Okay, I think this was really clear when we discussed genetic engineering, what is meant by it. We said that genetic engineering is technology that involves manipulating the DNA of one organism in order to insert the DNA of another organism. The inserted DNA of another organism is known as exogenous DNA. This is also, by the way, it is really important. So I have to mark it like this. This is really important. So I may ask you, write the scientific meaning. Exogenous DNA, you have to know that it is the inserted DNA of another organism. Okay, so clear and so easy. Then genetically engineered organisms are used to study the expression of a particular gene, investigate cellular processes, study the development of a certain disease. Aziz Muhayya, good morning.
Assalamu alaikum.
Khalid Habib, good morning.
Good morning.
Okay, so study the development of a certain disease, select traits that might be beneficial to humans. Then we moved to the tools. We said that genetic engineering can be used to increase or decrease the expression of specific genes in selected organisms. I'm just fast revising because all this exactly we passed it and it was clear for all of us. Who missed it or who want to understand it more deep, I think I send the link to you. You have the link of my channel. It is mentioned there. Also, I send it in the group. Okay. And organisms genome is the total DNA in the nucleus of each cell. I think this also, even I remember that I asked, I think it was Abdullah Ali, I asked him about this question last session. DNA tools can be used to manipulate DNA and to isolate genes from the rest of the genome. This is true. Restriction enzymes or proteins that recognize and bind to specific DNA sequences and cleave the DNA with that sequence. So there is a new DNA, a short copy of DNA. We want to connect it with the huge one. So there are restriction enzymes that, cut and bind. You understand? They are used as defense mechanism by bacteria against viruses. Okay, scientists use restriction enzymes as powerful tools for isolating specific genes or regions of the genome. I think this was really clear. This one example, EQRI, is a structure enzyme that specifically cuts DNA containing the sequence GAATTC, as we know. Sticky ends are single strands DNA sequences at the end of fragments can be reattached to complementary strands. I think I'm just trying to read it fastly. If something is misunderstood, just till the rest are coming. So now I have 10 of you. I still have 10 more. Where are they? I don't know. And so about the gel electrophoresis, we said that an electric current is used to separate DNA fragments according to the size of the fragments in a process. This process is called gel electrophoresis. When an electric current is applied, the DNA fragments move toward the positive end of the gel, and we discussed that. You will study it in organic chemistry to know what is the gel structure, where is the hydrophilic, where is the hydrophobic, where is the positive ball, where is the negative ball. Too many things. You will know it later, inshallah. The small fragments move faster than the larger ones. Unique pattern created based on the size of the DNA fragment can be compared to known DNA fragment for identification. By knowing a specific one, then compare it with a new one, you can know what is going on with it. Then we move to page 9. We'll discuss the DNA fragment from different sources can be combined to make new DNA molecules. Abdel Ilah.
السلام عليكم. Yes.
Abdel Ilah. Saad.
What?
Good morning, Abdel Ilah.
Good morning.
Mute yourself, please. Thank you. The newly generated DNA molecules with DNA from different sources is called recombinant DNA. So what is the DNA, the recombinant DNA? Hussein Al-Qahtani, can you please tell me what is the recombinant DNA, please?
Sure. Recombinant DNA is the newly generated DNA molecule with DNA from different sources.
Excellent.
So this is the new DNA that is formed.
Yes, wonderful. And exactly here is the proof of that. Do you see it? I think it is looking clear. Can you please, Khalid Al-Harbi, can you please, from this, you can please explain to us what your friend said now? I think
this illustration.
It is clear really in this picture, in this figure. Just look at it.
So the plasmid, the plasmid, it's open by itself and it's joined the fragments within A to make one whole circle.
So what is the function of DNA ligase? So what is the function of DNA ligase?
It's to join the two DNA. The new generated DNA with the different source DNA.
This is the recombinant DNA. Yes? Great.
Yes.
Thank you very much. Here, we have to know what is meant by gene colony. Sometimes you are going to the doctor that you have a problem with your intestine. I'm painful. I don't know. I didn't eat. When I'm eating, whatever I'm eating, I feel pain in my intestine. So doctor is making for you analysis, stool analysis, urine analysis, and CBC means complete blood picture. Then the result that there is just irritation. There is just irritation in your nerves, but he doesn't know. There is nothing serious to know. So he's asking, okay, we'll make a colony. So he is taking some of your blood, okay, like 5 millimeter of your blood, then making a colony. Here now we are discussing something that is related to it. Gene colony. Gene cloning. Let's come to read it together. To make a large quantity of recombinant plasmid DNA, bacterial cell or mixed with the recombinant plasmid DNA. Some of the bacterial cells take up the recombinant plasmid DNA through a process. This is called transformation. I think transformation means what? Means transfer something. Okay, the transfer itself. Transformation means, look here, if you look at this, this is the plasmid exactly, as you see. You look at it, there is a bacteria, transformed bacteria. Did you know this means transformed? Musa Haji, welcome back. Musa?
Yes, Mister.
Welcome back.
Thank you.
Can you please tell me, Musa, what is meant by transformed bacteria here in this picture? Omar, welcome back.
Maybe it's the combination of different DNA.
Combination,
the process.
combination
process.
of different DNA. This is the combined DNA.
Yeah, I mean the process.
The process?
Yeah.
You mean the process of the new DNA with all its combination? It is called the transformation. Do you think so?
I think so, maybe.
What do you think, Al Walid?
What?
What is what? What do you think
Sorry.
of what Musa said?
I think he's onto something, and I think that maybe it's sort of like a new gene, right? So that's why it's called transformed bacteria. So when it splits into another bacteria, that bacteria will have that new set of genes.
So the mom of bacteria, I mean the parent bacteria, it's transformed into a new bacteria. This is what you want to say, yeah?
Yes.
Okay. What do you think, Hussein, about this negotiation? What we are discussing, this discussion? What do you think? What is your opinion about that?
About transformation?
Yani, what I understand from what your friends said, because I have to know what they understood exactly. They said that transformation here, it is a process that a new kind of bacteria, transformed kind of bacteria, appeared from the plasmid DNA, the recombinant plasmid DNA. What do you think about that? Do you agree with them? Through
I
this
think
process
that,
which called transformation.
what you know about transformation is when a plasmid DNA enters a bacterial cell and then connect with the DNA, DNA to form a new, completely new or recombinant DNA.
So you mean that the structure? I mean, you mean that the bacteria is completely changed now. It was and became a new kind of bacteria. The characteristics of it is completely different now. This is what you mean?
Yes.
So you think that bacteriophage, I think you heard about it when I draw it for you in the class. I'll try to draw it now to maybe you remember it. When I draw it like this, then and here exactly as you know is the DNA, and exactly here it is something to allow it to inject. Yeah. Do you think this bacteriophage, for example, is a transformed bacteria? It was a kind of bacteria. Then by working with it, okay, and using this recombinant DNA technology, we changed it to be a bacteriophage. You know, the bacteriophage is really useful in our daily life in medicine. So do you think it was just, I'm asking your opinion, you and Mu'taz and Khalid Al-Harbi, all of you, do you think this is like the result of the gene cloning?
I don't think that it is the results, but it is the cause.
It is the cause, yeah? So it is
Yeah.
not a result, it is just causing
The
it.
in a sequence.
The DNA sequence. Okay, that's exactly what I want to hear from you all to be sure that you started understanding why I am teaching you this chapter or this lesson especially. Because by understanding this, you will look, maybe you will change the image of your future. One of you is looking, for example, that he preferred to be electronic engineer. Okay, but I think when he's thinking about this, okay, why not to be biological engineer? Why not to think and to come deep in these details? Maybe I can create something in the labs that with much benefits to the humanity. So this is something I'll obtain a great reward from Allah subhanahu wa ta'ala, then great money, huge amount of money you can get from um, your government. That's something just I want you to think well about it, honestly. Okay. Look here, please. Abdullah Al-Eid, yes.
Yes. Do you know any university in Saudi Arabia that studies the genetical engineering?
Yeah.
Assalamu
Alaikum assalam. Who is that?
Khalid Sutton.
Yes, Khalid Sutton. Welcome back, Khalid.
Hello, welcome, sir.
Khalid Sutton. Okay. What's wrong in this, Abdullah?
No, I want to know because I tried to find, but I couldn't. I didn't know.
Habibi, you know, to enter this, it's not like you have to enter this college, so you are entered it, that's it. No, it's not like this. First on all, you have to pass, you know, the first steps, the exams of accepting and all what you know exactly about it. Then, when you are entering it, you have to study the first year and the second year and the general science. Then from the third year, you are going to be specialized in biology, in chemistry, in biochemistry, in microbiotic, in organic chemistry, in inorganic chemistry, in atomic chemistry, in whatever the field you prefer. You got the idea? So there is one branch, it's called DNA technology. Only DNA technology. You got the idea? So I think this is steps. It's not like you want to go here,
Yes.
so it will be easy to go ahead. No. It is exactly in Al Haras University, available in Al Haras University. It is in Saud University. You understand?
Yeah.
It's not only in this kinds of this foreign university. Yeah.
Because
Okay.
I think it's very interesting to learn
Yeah,
about
it is
genetics.
amazing. It is really a
Cloning.
hard subject that what you are studying, and it is dangerous if you are not 1000% caring about yourself and your health and using all the steps of visiting the labs. But it is really something so interesting.
Yes.
Yeah. So bacteria that take up the plasmid make copies of the recombinant DNA during cell replication. This is easy. Large number of identical bacteria containing recombinant DNA can be produced through this process called cloning. So the cloning, actually about cloning, it is one chapter, complete chapter to know what is cloning, the steps, how to do it, what to use, what tools you have, and everything, and especially that we are studying online only. I couldn't help you in the lab because there must be a lab and show you some tools. And even I asked the administration to bring these tools for me to show it to you. So I have to delay this part, especially, but I promise, inshallah, if the situation in the world will be better about COVID-19, I promise I'll ask for this and do it in the lab, inshallah, next year. I hope that, inshallah. Okay, the DNA sequence. Scientists study DNA sequences with DNA fragments, DNA polymerase, fluorescently labeled nucleotides, and gel electrophoresis. Also this, I have to delay it because this exactly we will do it, I swear. I promise you that we will do it in the lab, inshallah, but step by step. Polymerase chain reaction. Let's come to consider this, it's really important. Once the sequence of a DNA fragment is known, a technique called the polymerase chain reaction, PCR. I think now you know what that does mean, PCR, what the word is speaking. Did you make the PCR? No, so we don't know that you are infected by coronavirus or not. We have to be sure that you're infected or not by PCR. What? We came to a very important point in your daily life. So what is PCR? A polymerase chain reaction. Can be used to make millions of copies of a specific region of DNA fragments. This is true. So by increasing the amount of it, we will know, because maybe a small amount of this virus or this bacteria cannot be appeared, cannot be recognized during the tests. But to increase its amount, it is easy to know it exists in the blood or not. PCR can copy or amplify a single DNA molecule numerous times for use in analysis. Mu'taz, is
Mr.
it clear for you, Mu'taz?
Yes, Mr.
Wonderful. Yes, Abdullah Al-Eid.
You said if they want to find the coronavirus in a body, and if it's too little, they should multiply it to find it, right?
You are taking some of the blood. Then they are taking one of this, not cells. You know, it is producing protein in the blood, because coronavirus is specialized, very specialized. So it is going only to the lungs, the lower pulmonary, as you know. So, but there is reaction in the blood. There is immunity, defense are going to attack this virus. So they are taking this immunity because they know exactly how it is looking like. The immunity of this coronavirus should be shaped like what. So they are increasing its amount, then in testing it to know it is exists or not. If it exists, so it is infected by coronavirus. If not, so okay, what is this? They are searching because they have copies of coronavirus, they have copies of HCV, they have copies of HIV, they have copies of many, many different kinds of viruses. So they will know what is it. You got the idea now, Abdullah?
Yes, but why don't they just use PCR to multiply the immune cells against?
Because I told you there is S spikes on coronavirus, so it is making mutations so often. So you don't know which one of them, which copy of them it is that is attacking the trachea, which one is that is attacking the lower pulmonary, what's attacking the liver, what's attacking the heart, what's attacking the kidneys, what is it can be infected by the air only. What's really, it is, you know, it is worse than cancer. It is worse than cancer. Believe me. You got the idea now?
Yes.
Yeah. Any other questions?
No, thank you.
You're welcome.
Mr.
Khalid Al-Harbi.
Can the coronavirus transfer from person to another from the air?
Yeah, this is the 9th copy of it now. You can imagine it mutates itself to be transferred by the air. This is why here in the wise government, what we have here in Riyadh, in Saudi Arabia now, they said that please sit at home for the 24 hours. No way to go outside. No way. They know what is going on.
Mr.
Yes.
Is PCR easy to do? Simple?
For you, it is not easy because you need tools for that. But I'll do my best next year, inshallah, if we'll be together, inshallah, next year, and Allah will save us from this pandemic disease, what we have in the world now. I promise, inshallah, I'll do my best and even more than my best to make it in the lab. Okay? But we'll make it on a rat or on, for example, on a rabbit. Okay?
Okay.
Okay. Now, biotechnology is the use of genetic engineering to find solution to problems. If that's true, because you know nowadays all over the world they are concentrating with the biotechnology labs to see what's going on, what to do with this coronavirus, what to do. Yeah, so they are working on this field. Organisms with genes from other organisms are called transgenic organisms. So it is called what? Transgenic organisms. So Rakan El Bays, can you please tell me what is meant by transgenic organism? Mr. Rakan,
Yes, Mr.
can you please tell me what is meant by transgenic organism?
Um, I think transgenic organisms. I don't know, Mr.
Okay, no problem. Can I ask Khaled Satan to help him? Khaled Satan. Khaled. Okay, Nasr Sharif, can you please help your friend?
Okay, what is the question?
The question is
Again?
what is meant by transgenic organisms?
Okay, transgenic organisms are organisms with genes from another organisms.
From other organisms. This is you are telling me or telling to Rakan?
What?
You are just discussing this with me or with Rakan Khalid?
With all of us.
No. Speak to Rakan because he wants to know. He told me I don't know, so I asked for any volunteers, and you agreed. So please help him because I want to listen from him. I want this answer from him.
Ah, okay. If you look at the screen, it's in red. Transgenic organisms.
Yes.
So Rakan, do you understand now what is meant by transgenic organisms?
Yes. They are organisms that are that have been altered. No, that are from different.
So it is the organisms with genes from other organisms. So
Genes from
other organisms. I think we discussed it the last session when I told you we have in Saudi Arabia. Saudi Arabia is famous about sheep. Look at, for example, Australian sheep. Australian sheep has too much meat. So what is not to take this gene that allow the muscles to grow there and insert it in our new generation of this Saudi Arabia sheep. You understand? So
yeah.
this gene that is mentioned with this organism is called transgenic organism. You got the idea now?
Yes, I guess.
Is it
So
clear?
Ashraf,
Yes.
are we now currently able to lend some genes to another organisms or not yet?
Yes, we can. We do eat many in the plants.
What about
We
human?
do eat
What
many in
about
the plants and in some animals right now.
yeah?
Okay. Transgenic animals, plants, and bacteria are used for research, medicine, and agriculture. I just told you plants means agriculture. You got the idea now? Great. Transgenic animals. Oh my God, let's come to read this about transgenic animals. It is something really looking interesting to all of us. Scientists produce most transgenic animals in laboratories for biological research. They are used to, this is very important, please, dear students, this is really important. All what is read is really important. They are used to study diseases, improve food supply. What I told you now about improve food supply, I told you about the Australian sheep and Saudi Arabian sheep. And how can we make a hybrid of it? Okay, improve human health, be potential sources of organs for transplant. Okay, this is really I want you to consider it because it's really important. About plants, frequently genetically engineered for resistance against insects or viral pests. Here, I remember something exactly. I made investigation about it. It is a kind of viruses that infecting potatoes when it is growing, potato plant. When it is growing, it is making it, it is like white spots appeared on the potatoes. These white spots, when you see it in one potato, stop eating it immediately because it's really cancerogenic. So, and the raw potatoes, I mean. So, the science, I remember that very well. So we do what we made a research, but exactly I finished with my college before finishing with it, but I was considering it. They exactly found a transgenic plants of potatoes that can be stop this virus to attack it. This is why we have potatoes now without these spots. Understand? I think now you don't remember that you see potatoes with these spots nowadays because it is over. But for me, I remember it because I suffered from it really long ago. Okay, so other transgenic plants are designed to reduce allergic reaction in humans. I think you know what this means, allergic reaction. Reduce the histidine, the percent of histidine in the blood. Contain increased vitamin and mineral content. This is true. Resist extreme weather. Yes, they can stand in this extreme weather, especially here in Riyadh. Produce vaccines or biodegradable plastics. Subhanallah, they can do that. Okay, about how and what is going on exactly, step by step. About bacteria, transgenic bacteria can produce insulin. This is easy to do it even in the lab. Very easy to do it in the lab, inshallah. Inshallah, I'm planning for that. Okay, produce insulin and growth hormones. Slow the formation of ice on crops. Clean up oil spills, like if something happened exactly in the ocean, like this one oil ship, something is drawn or something like that, then oil is floating. How to solve this big problem? What is going on? Everything exactly, it is becoming easier with this kind of bacteria. Decompose garbage. I think now you see that the garbage is even for sale now. The garbage, there are companies are coming and collecting it and decomposing this garbage to produce, for example, methane. Methane is a gas that you are using at home for your gas. Okay, so all of this exactly, many things, what you will enjoy it. I'm sure that I finished with my session now. I hope that you really was interested to this session. I hope from Allah that you got the maximum benefits of this session. So now I want to ask if one of you had any question about what we studied. I'm ready to listen to it for 2 minutes. Abdulaziz Muhayya, everything is okay for you? Abdullah Al-Eid, everything is clear for you?
Yes, everything's clear.
Al-Walid, clear?
Clear.
Clear?
Clear.
Yeah. Fahad Al-Awaji, clear? Qahtani. Qahtani,
Yes.
everything is clear?
Yes.
Khalid Harbi.
Yes, alhamdulillah.
Wonderful. Khalid Habib. Khalid Satam.
Yes, Mr. Yes.
Okay. Khalid Satam, everything is clear? Mu'taz. Musa.
Yes, thank you.
Nasser.
Yes, Mister.
Omar. Rakan al-Biz.
Yes, Mr.
Alhamdulillah, that's something is cheering me up. Okay, I'm happy that I heard from you all, and I hope to continue, inshallah, next session. See you then, and goodbye.
Bye bye.